Great, so thank you. I'd like to thank the organisers for giving me the opportunity to talk here about a slightly different dark archive, but that we hope to explore and reveal through the new and emerging field of biocode ecology. So, what is biocode ecology? Well, it's the study of the biomolecular information that forms part of the biological history of manuscripts. So it's the biomolecules that are trapped on the surface of these manuscripts that can reveal something about their history. So there are two main biomolecules that we're going to focus on. The first is proteins. So almost everything organic that we see is predominantly made of proteins. They are ubiquitous, they are everywhere. So they're a very good molecule to target. We can go by this through two different processes. The first one is that we look at one particular protein. It's called a single protein analysis, or also peptide mass fingerprinting. Now, in the case of manuscripts, we're looking predominantly at parchment and also at a small level at leather as well. But both of these come from animal skins, and the dominant protein in skin is collagen. So by looking at this collagen protein, we can actually reveal the species of animal that it's come from. So what species of animal have been used to make the parchment or the leather. If we take another approach and we actually do a proteomic analysis, this starts to look at all the other proteins that are surrounding on the surface of these documents. And here we can start to think about surface treatments that we use to prepare the parchment, or even things like stains or what's going on through the handling and conservation. It's also very useful because apart from identifying, let's say, the presence of wheat, for example, or peas, which things that we have found, proteins are tissue specific. So in this case, for example, we could actually distinguish if we find egg yolk from egg white, which can actually be very revealing. The other biomolecule of interest, and the one that most people have heard about, is DNA, which we also look at. In the case of genetic analysis, we can also get the species of animal from the parchment or leather, but we can also go a step further and we can get the sex of the animal, which can be hugely important for many reasons, as we'll see later. And in some cases, depending on the quality of the genetic data, we can also get a sense of relatedness. So perhaps we can identify if animals belong to the same flock or herd, which can be very useful for provenancing. Apart from the animal DNA, there's also other levels of DNA that are stored on these manuscripts, and one of the important ones is the microbiome. Now, this is the collection of bacteria and microorganisms that live basically everywhere, but obviously on manuscripts too. It can be hugely informative in the sense of looking at a sort of the environment that these books have lived in, how their handling and storage and conservation may have affected it, and we'll see examples of this later. Finally, we can also look at the human DNA, although I have put this in a slightly lower position because we don't tend to look at it as much. It is true that we do see human DNA, but it's much harder to actually get useful information out of this, because obviously the human DNA has built up over time and will be a complete mixture of all the people who may have used it, handled it, produced it. So for our line of work, it's not as important, but it is there. So how do we actually go around about recovering these biomolecules? Well, we use something called triboelectric extraction, which is an incredibly fancy name for we use an eraser. And it really is a simple technique, and it really is just using an eraser. It's based on the electrostatic effect, which is what you can see represented here with this lovely Wensleydale sheep. And very simply, we take individual pieces of PVC eraser and we gently wipe it on the surface of the parchment. So we're not talking about harsh rubbing, we just simply wipe it on the surface. And we collect all the eraser crumbs that come off. So normally the stuff that you would throw away, that's what we collect. And it's from those little eraser crumbs that we can then extract our biomolecules to do our further analysis. If you are interested in knowing a bit more about the technique and the whole process, we do have a video on YouTube. If you put in parchment zooms, and it will give you a nice explanation of how the whole process works. So, depending on the amount of eraser rubbings that you have, you can perform different kinds of analysis. For our protein analysis, as you can see, we need very small amounts of eraser rubbings. Bearing in mind these tubes are about this size. And that is the analysis we have done most frequently. To date, we've done over 5,000 protein analysis from manuscripts. If you want to look at the animal DNA, we're talking about a considerable more amount of eraser crumbs, and that is not always possible depending on the status of conservation of the manuscript, but it is that is ideally what we want. We have worked with less, but that would be the ideal. And then if you're only interested in the microorganisms, it's somewhere in between. So not as much as for the animal DNA, but a bit more than proteins. So once you have your eraser crumbs, you have to obviously extract these biomolecules. It's a slightly different process depending on if you have proteins or DNA, but very essentially you are going to extract them in a saline solution, you are going to clean them up, and then you will do, in the case of DNA, genetic sequencing, or in the case of proteins, we use mass spectrometry to be able to identify proteins. We've recently published a paper that details all the different processes and how you can go about choosing what method is right for you in heritage science, which you can see here. So I'm going to give you some practical examples of where we've actually used these techniques and what they've actually helped us reveal about some of the manuscripts. I'll start with the protein analysis. So like I said, it's a very, very simple technique. We recover our razor crumbs. We heat them in a saline solution to pull off these molecules. However, in order to do our mass spectrometry analysis, we do need to cut our protein first. So I've said that the protein we target is collagen because it is the predominant protein in skin. Collagen is an incredibly long structural molecule, and we can't analyze it in its complete form. So we do have to cut it into smaller fragments. So we have to add a protein called trypsin to cut it into smaller fragments. But it's important that this protein only cuts in fixed positions. So that's going to then help us decipher what these fragments are going to mean. After that, we really just clean up the sample to make sure there's nothing interfering. We spot tiny amounts onto metal plates and let them dry, and then we perform our analysis. So the analysis that we use is called MALDI-TOF. It's a very long name, but the important bit is the TOF, which is time of flight. And this is the basis of that kind of analysis. So a mass spectrometer is quite big. It's bigger than this table. But essentially, it's empty. Inside is a long tube, and it's a vacuum. And what we're going to do is we're going to put our little cut-up fragments of our protein into that tube, give it a kick of energy, and they will travel down. And depending on their size, obviously the ones that are smaller will travel a lot quicker. Those that are larger will take a longer time to travel down the tube. And by measuring at what point they reach the detector, we can discriminate different sizes. Now, in this example here, as I've said, collagen is a long molecule. It's incredibly conserved in all species. It's such an important protein that it can't allow for a lot of mutations. However, there are very small differences. So you can see in these two examples, these colours represent the sequence of the collagen molecule, and there's only one difference, a green at the top and a grey at the bottom. But this accounts for a mass difference. It makes that fragment a little bit heavier or a little bit lighter. And it's by looking at those little tiny differences that's going to allow us to discriminate, for example, in this case, between sheep and calf. So this is what we actually get out from our machines. These are our spectra. But essentially, we get what the theme is called is peptide mass fingerprinting. We get a fingerprint for that protein. So although they look very similar, these very small adjustments, which I've just shown, allow us to discriminate between all these different species. So it's a very simple way of being able to identify, in this case, parchments. So we applied this analysis to a 12th century gloss Gospel of St. Luke that was produced in Canterbury. It's held by a private collector, and it's in its original binding. The main text is written by 2 scribes. The glossing, I think, is about 5 they identified, and it was listed as having 158 vellum folios, and there was no description of the binding per se. So we decided we were going to do a complete analysis of this book. So we took samples from every bifolio and from the bindings, and what we thought would actually, going on the assumption that it was just going to be calf, this revealed that we actually had 5 different species of animal in this one book, which is really exciting for us. So we'd identified roe deer on the binding, the strap of the binding is actually a fallow or red deer. And then inside, in the quires, we have calf parchment, sheep parchment, and even some goat parchment. It seems to be laid out in quite an organised fashion, at least the first half of the book. So they're interchanging calf and sheep parchment. There is a quire where they get it the wrong way round. But then there is a change. They go I have a complete quire of just calf. They start to then introduce one bifolio of sheep. Then there is some goat, which we really cannot explain. Also, it's interesting to note that the goat is not of good quality. It's quite tick-marked. It's not a particularly nice piece of parchment. So why it was put in there, I'm not quite sure. The interesting part comes in the subsequent quire, which goes back to the original structure of calf, sheep, calf, sheep. However, on the first folio of that quire, there is a change in scribe. So here we've had the main scribe writing the main part of the text, and here it changes to a second scribe, which, from the experts who have looked at it, have commented that the second scribe actually wasn't as good. His hand was not as nice as that first scribe. And it's interesting to note that as of that point, all the quires then turn into sheep. And sheep parchment is generally considered to be of inferior quality to calf. So whether that is significant or not, we don't know, but it certainly is, it's remarkable that it happens at that specific point. Apart from identifying the species of animal, with our mass spectrometry data, we're also able to get an idea of what we've called parchment quality, but this basically measures a very specific kind of damage that occurs on the collagen molecule. And we think this damage occurs during liming. So when you prepare your parchment, the first thing you need to do is put it in lime to dehair it. Now, lime is an incredibly alkali solution. So if you leave it there for too long or it's exposed to a lot of alkali, you will get a very particular type of damage called deamidation on the collagen. And it's that we can measure. We can measure this damage. So the more damage that these documents have, the lower their quality index that we give here. So a higher exposure to lime, shall we say. And those with higher, obviously, are the opposite. So we did these quality indexes for all these bifolia. And what we could see was there's starting to be some groupings, especially in the calf parchment. The fact that these different bifolia had a very similar quality indexes seemed to indicate that they could possibly come from the same skin, because a skin would obviously be treated as one batch and would have similar properties. So my colleague, Yuji Vanucek, who is a conservator at the Royal Library of Copenhagen and who I did this project with, he looks at the visual aspects of parchment to identify the animal and the anatomy. And he was able, by selecting these particular bifolia that grouped together, he was able to reconstruct the skin of the animal from these bifolia. These are images taken with transmitting lights, which for him are the best way to be able to see the anatomical features of the animal. And by being able to recreate the skin of the animal, we were able to get the dimensions of the animal. And interestingly enough, it's exactly the same size as this statue that we have in my department at York. So this case, which is a happy coincidence, but just gives you an idea of what a parchment calf would look like. And we certainly know that they are talking about calves from between 2 to 4 weeks, and certainly no more than 6 weeks, because their skins become too thick to produce parchment. Now, to give an example of the DNA analysis, I'm going to be focusing on the York Gospels, which we were kindly given the opportunity to analyse not long ago. The York Gospels, are an Anglo-Saxon gospel book produced in Canterbury. They were brought up by Bishop Alston to York. It's still used today, although not all the time. But it's on exhibit in York Minster. So again, we wanted to do a complete analysis of this book, so we did all the bifolia. This one was a slightly more predictable story. It was indeed all calf parchment, the Anglo-Saxon part, with the exception of one bifolia of sheep here, which nobody seems to provide an explanation for. We don't know. We don't know if they didn't realise or if it's intentional, but everything else is calf. However, we were able to identify some sheep folios at the beginning of the document. And this is a later edition from the 14th century. So it was nice that we were able to pick up that, not only from, obviously, the records, but it had a biological significance as well. These folios at the end that are in grey, we were not able to get an identification for, and that's because they were heavily conserved in the early 20th century, and they basically have a silk gauze pasted all over them, so it didn't allow us to actually pull off any biomolecules. But apart from doing our protein analysis, here we actually decided to do some genetic analysis. Now, we can't do the same number for genetic analysis for reasons of cost and time. So we took a subsection. We took about 8 random folios to look at. And we did various types of analysis on them. So the first thing you can see here, these are called PCA plots. And here what we're looking at are modern cattle distributions globally. And what you can see is that our samples from the York Gospels cluster in this area, which correspond to European cattle, so as we would expect. If we then look at this in a bit more detail, we start to pull out the European breeds, we can see that, again, our samples seem to cluster a bit towards northern European animals, but don't fit perfectly. Now, this is something to be expected because, of course, we're talking about medieval animals, and here we are comparing them to modern breeds. Even if they are so-called rare breeds, these are not authentic medieval animals. So to compare them with medieval or older data is obviously going to be problematic. In an ideal world, you would have a comparative data set of older animals. But they certainly plotted towards Northern European. The other interesting piece of information we were able to get was the sex of the animals. We were able to get the sex of 5 of them. I won't put 4 here, but there are 5. And of those 5, we found 4 of them were female and 1 male. And although this is a very small subset, it is interesting to note that there was a predominance of female because we had expected to find more male. We had hypothesized that if you have a dairying economy, you would be keeping your females to maintain your herd, and you would only have a few males to carry that on, therefore culling the rest of your males. But what we find here is the opposite, so that was actually quite revealing to us. The other aspect that we were able to look at was the microbiome, so the microbial signature that lived on this manuscript. Now, many of you may have heard of the Human Microbiome Project, which is a project to categorize all the different bacteria that live on the human body. So depending on the different parts of your body, you have different groupings and families of bacteria. So we plotted our data from the York Gospels, comparing it to some of the human microbiome data. You see here, this is stool bacteria, mouse, and ours plotted very nicely with nose and skin bacteria, which would make perfect sense because what we're picking up is a signature of people actually using and handling this document. We actually then did a little bit more, and we started to look at the kind of the types of bacteria, or the families of bacteria that you could find on the different folios. So what you can see here in blue, these are the original Anglo-Saxon folios. And you don't have to see what the type of bacteria are, but if you can just have a look at the colorings, you can see that they seem to exhibit a very similar profile. So they all seem to have a similar bacterial component. If we then look at the pages here in orange, this is the later 14th edition that we identified as sheep. Now, although it has a similar profile to the Anglo-Saxon, you can start to see differences in proportions and some different families starting to appear. Here in red, now this is the folio that I said we weren't able to identify because of the conservation, but it has a completely different microbial profile, and this would make sense. We're only seeing post 20th century intervention there, so it gives it a completely different microbial signature. And then just for comparison, here we have a set of 6 documents from the Borthwick Archive where we developed the sampling technique, and you can see again they have very different bacterial compositions. So although this is only one example, it offers the possibility of actually using these bacterial signatures as maybe another form of tracing documents. It's another internal fingerprint that these documents carry, and I think are worth exploring. Can maybe tell us something about the production, where it's been how it's been handled, where it's been stored, and any conservation work that's gone on. And finally, just to compare these two analyses, because we often get asked, can you do DNA analysis on such and such? And people often forget about the proteins. There is a difference in time and resources, and the questions you can answer with each of these techniques. So it is important to think about what it is you're trying to find out and what's the best approach to get to that answer. Protein analysis is a lot cheaper and a lot quicker, and we can do a higher number of samples, but as you've seen, it has different limitations on the information we can get. DNA analysis can provide potentially more information, but is a lot more expensive in both time and money. So we really need to think about how we approach, if you want to answer some of these questions, how we go about tackling them. And finally, I'd just like to thank our collaborators who've kindly donated us a lot of samples and without which this work wouldn't have been possible. Thank you for your time.
Thank you, Sarah. Excellent timing. We do have time for a few questions to Sarah. Yes, that.
I have a question about the animals used for the bindings. Is there something like, I don't know, database about cases that exist that can be explored?
So if we have analysed it and we have published it, that data is deposited in public depositories. You can go and interrogate it, and the answer is we would like to share all this data, but we don't know how yet. So the data belongs to the institution. We have a collaborative arrangement, and it's up to them what they do with it. We would very much encourage that they add it to their catalogue entries. And anything that we have deposited and that has a DOI, we would love to link with catalogue entries so this information is available. But we don't have a complete sort of system that we can put out there yet.
Did that answer your question about bindings? Okay. Yeah, Carolyn.
I've read about cases in which archaeologists use isotopes to determine the original home of people from archaeological findings. Do manuscripts contain any material that could be used for this sort of analysis?
So we actually had a PhD student do exactly that. There are two problems with this. The first one is for isotopes, you do need destructive sampling. So that does mean cutting fragments off manuscripts, which unfortunately obviously is not a case. We were able to do it with pieces that were donated to us from more modern samples. And the answer to did it say anything is not really. It gave quite a confused signal, so clearly there is other interference there. The fact that it's skin and not bone, the turnover of the protein is not the same, so it didn't give a clear-cut answer.
Mark, yeah.
What sort of disciplines do you need to have in a team that does?
We have a bit of everything. So we have, I'm a biochemist. My colleague who I collaborate with, Matthew Teasdale, is a geneticist. Yuji Vimicek, who I mentioned before, is a conservator and experimental parchment maker. We work with archivists, we work with curators. We kind of need to work with everybody because it is very much a collaborative effort. We do not want it to be just we stick this in the machine, give you a result, and that's it. We want to bring in people from all the disciplines from the beginning to discuss what is it, what are the questions, how can we answer them, and we seem to have done that till now, and it's worked really well.
And one of the key points, isn't it, is that Sarah was talking about the rubbings, the eraser crumbs, and the crucial thing about that is that that's the byproduct of conservation. So it's not necessarily something that you're doing for this, so you can do it for that. It was an observation that I think Sarah made originally when she watched conservators at work. She noticed them using these plastic erasers and all of these crumbs falling off, and that if those crumbs contain enough of the protein molecule to be able to analyse. So it's a beautiful example of the byproduct of something else, of a process that was happening anyway, to provide a whole set of new information.
Exactly right.
Yeah. Well, I think you missed that bit, so I thought I would just add that in. Any other questions? Yes.
Can you send us to ink for pigment? I mean, I don't know if I want to read.
No, this wouldn't work because ink that would be organic, etc. But you can. The best way to be for interrogation of inks, things like Raman and XRF. I know the team at Durham University Team Pigments, they're very hot on this, so they've been looking at inks from a whole range of manuscripts. So yeah, they have other methods, but we wouldn't find proteins or DNA there.
Oh, I was actually going to ask about glue, so I guess that's about glue.
Yes.
Glue.
So that's something we have been asked to do. We're currently looking at that. Certainly, we have had some cases where we've analysed parchment and it's come back as sheep and cow, for example. Not all cow, no, sheep and cow, both are there. And usually we've gone back to people and said, hang on, is there something here? Was there a glue? Was there an adhesive? And in nearly all the cases is yes, there was an adhesive used here. So we're certainly able to identify collagen-based glues. We haven't tried, for example, looking on paper, things like sizes, but that's another step that we're currently looking at doing.
Yeah, Sue.
And I think one of the things that's sort of thinking about in the field is you're working on wax as well.
Yes, so we have an extended project called Archives, and we're looking at the biomolecular signal trapped in wax seals. So I have colleagues who are now working on exploring to try and extract proteins and DNA that is trapped when seals are actually made, and what that information can provide about the people who made the seals, used the seals, etc. So that project is literally just starting. So
People who made the seals and used the seals, but also the bees
yes,
that made the wax.
Yes, and the health of the population of the bees.
Population.
Yeah.
So there's wax that comes from Eastern Europe, and also wax that comes from North Africa, and how that is being. So the whole sort of bee economy as well. In your field. Yeah. And then there's also the potential for very interesting overlap between the species identification that's happening through the laboratory-based science with species-based identification through high-resolution digital imaging. And it's when you can get those two working together that we can start to see this on a macro scale. And that's when you can start to get to the economy, the monastic economy, and beyond. And seeing these books as herds of animals sitting on the shelf.
And dated.
Dated
for us
herds of animals.
Hugely important.
Yeah. So as an archaeological resource, manuscripts are now opening up new avenues for archaeologists to think about as biological data as well. And one of the, I've been working quite closely with Sarah and Matthew Collins and their team, is that as scientists they're tremendously interested in the potential of these archives for applying and developing and enhancing and improving their resources. But they also want to know what are the questions we want answers to. So they're a very responsive team to particular historical problems that you might have.
Which is why we really want to include anybody who would like to collaborate on our project. We welcome people from all different disciplines to come in.
Because this is an example of extraordinary new technologies that we can apply to our data, but we don't want to apply them just because we can, just because they're there. We want the analysis to be driven by major research questions that enable us to do things differently that we couldn't do in other ways. Okay, Sarah, thank you very much.